Thank you for virtually joining us for the latest version of the Variant Effects Seminar Series. My name is Diego, and I'm a member of the VES Organizing Committee, along with Jan Ilbudu and Adrini D'Souza. These seminars are made possible with support and a lot of help from Lara Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a huge thank you to them. Also, we are looking to add a few more people to the VES Organizing Committee. So if helping run a seminar series seems interesting to you, then please follow this link to find extra information about applying, and I'll share it in the chat in a little bit. As a reminder, we are also always on the lookout for potential speakers for future seminars. If there's anyone you'd like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting the presentations, I just have a couple housekeeping items to mention. Each talk is about 20 to 25 minutes, so there should be time for some questions. Please post those questions at any time to the Zoom Q&A, and then we'll read them out to the speaker at the end of the seminar. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can catch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. Again, I'll share this in the chat once we get started, the URL. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. And for the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. So without further ado, we have two great speakers scheduled for today's presentation. William Bakash is a postdoctoral fellow at the Quantitative Virology and Evolution Unit at the Laboratory of Viral Diseases under the supervision of Patrick Dolan. William is developing approaches to explore the mutational landscape of viral proteins. In his talk, William is going to share his unpublished work related to investigating the tolerance of a variety of insertion sequences in viral proteomes. The title of the talk is Deep Insertional Screening Reveals Hotspots for the Insertion of Useful Tags in the Non-Polio Enterovirus EVA71. So William, go ahead and share your screen and start whenever you're ready.
Hey, can everyone hear me well?
Yep.
Awesome. All right, I'm just going to remove all these small blocks from here. Thank you, Diego, for this introduction. I would like to start by thanking the Atlas of Variant Effects Alliance for allowing me to present my work. This was really one of the first seminar series that I started joining at the start of my postdoc, and so it was instrumental for my work. And so, like Diego mentioned, I work at the Quantitative Virology and Evolution Unit, where we study RNA virus evolution using quantitative methods. So I've changed, sorry, a bit, the title of my talk, and it will be today, Uncovering Enterovirus Structural Plasticity Through Deep Insertional Screening. I would like to start by a general introduction on the importance of insertions in shaping RNA virus evolution. In this hypothetical phylogenetic tree, we can see this ancestral virus, which has a certain arrangement of domains. However, throughout evolutionary time, this ancestral virus will then diverge, leading to the emergence of new modern viruses that have different arrangement of domains. And this can be due to either a loss of domains or a gain of domains through insertions. And while we can sample pretty well modern viruses today through techniques such as metagenomics, we are still lacking in our ability to predict virus evolution in the future. Furthermore, we still are not capable of fully understanding the constraints of viral genomes for insertions. And the process that I'm describing here happens at a much faster timescale for positive single-strand RNA virus genomes. And this is because they have higher mutation rates and recombination rates. An example of positive single-strand RNA viruses are enteroviruses. Now, these belong to the picorna family of viruses and cause diverse human diseases. The enterovirus that I'll be focusing on today is called EVA71 and is one of the main causes of hand, foot, and mouth disease. On the right here, I'm showing you the different outbreaks of EVA71 throughout the years and the probability of death or hospitalization due to these outbreaks. To note that the EVA71 strain that I'll be using today is derived from the outbreak that happened in Taiwan in 1998. Focusing a bit more on the EVA71 genome organization and life cycle, similar to all enteroviruses, EVA71 is a positive single-strand RNA virus. And therefore, when the viral genome enters a host cell, this will be directly translated into a large polyprotein. Now, this large polyprotein can then be sequentially processed by a viral protease, yielding to these individual viral proteins. And these viral proteins can then be divided into two groups: the structural proteins, which leads to the formation of the outer shell of the virus, otherwise known as the capsid, and the replication proteins, which will form at replication membranes, leading to the formation of the replication complex of the virus. Now, the constraints on evolution on these viral proteins depend on their individual functions, but the constraints also apply to their interactions in these complexes. And proper assembly of these complexes is obviously very important for the virus life cycle, where proper assembly of the structural proteins is important for entry into a host cell. Similarly, proper assembly of the replication proteins is important for optimal genome replication. Now, going back to constraints on viral evolution, what information do we have on the role of insertions throughout enterovirus evolution? And to illustrate the importance of insertions in shaping enterovirus evolution, I'll be giving one example specifically on the enteroviruses, rhinoviruses. Now, here I'm showing a multiple sequence alignment of rhinovirus A structural protein and rhinovirus C structural protein. And we can see that rhinovirus C structural protein contains a 12 amino acid insertion. Doing structural comparison on rhinovirus A and rhinovirus C structural protein shows that this insertion leads to the formation of this finger-like extrusion. Then, when we compare the morphologies of rhinovirus A and rhinovirus C, what we can see is that rhinovirus A has a smoother virion surface. However, rhinovirus C has a spikier appearance due to this finger-like extrusion. Now, this also leads to differences in receptor usage, where rhinovirus A uses the ICAM1 glycoprotein for entry, and rhinovirus C uses the CDHR3 glycoprotein for entry. And so we can see how insertions can lead to changes in molecular mechanisms of entry of the virus. And this has subsequent important effects on the pathogenesis of these different viruses and also on the symptoms that they cause in patients. However, this is just one example of an insertion event, and constraints of insertions over the whole viral genome remains poorly understood. And this is where deep mutational scanning studies are very powerful, because they can measure the fitness effects of many mutations in one experiment. And so here I'm presenting the only real deep mutational scanning study on enteroviruses. Specifically, these authors focused on Coxsackie B3 virus, where they measured the mean fitness effect of all possible non-synonymous mutations over the viral capsid proteins. They were able to see that the sequence entropy or diversity of these regions in nature correlated well with the mean fitness effects measured. And this shows that certain laboratory experiments can sometimes replicate what is happening in nature. However, these measure only non-synonymous mutations, and the effects of insertions are not represented in this type of data sets. And so this takes me to my main goal here, which is that deep insertional scanning studies are needed to investigate fitness effects of insertions in enterovirus genomes. Leading to the objective of my work, the goal is to explore the tolerance of the EVA71 proteome to insertions. We want to ask where are insertions tolerated in viral proteins, and how is that tolerance affected by the size and the amino acid composition of insertions. The second main question is, where are these tolerated insertions localized on the structures of these viral proteins, and if these regions are conserved throughout viral evolution? And to answer these questions, we'll be using the technique called deep insertional screening, where we will take an insertion sequence of interest and insert it in frame into the viral genome. This will lead to the generation of this plasmid library with an insertion sequence at every possible position in the viral genome. And then we can take this library, rescue the virus, and this will be our subsequent selection method, and then sequence the output. And then this will allow us to measure the fitness effects of insertions on viral genomes. And here I will start by describing the methods needed to generate a deep insertional plasmid library. And so we'll be using a pipeline which is called Saturation Programmed Insertion Engineering, otherwise called SPINE. This pipeline was developed by Willow Coyote Maestas, and I want to thank him first for developing this pipeline and also for being helpful when asked questions. And so for people that are not familiar with this pipeline, it has two main aspects. The first aspect is a synthetic biology aspect, where the diversity introduced will be coded by these synthetic oligopools, and this allows us to generate balanced and diverse libraries. The other aspect is the Golden Gate cloning aspect that will allow us to clone these synthetic oligopools into a destination vector. This Golden Gate cloning is the preferred method because it is very efficient and allows us to build large libraries. Now, talking more into details about this mutational engineering pipeline, we've adapted it to a viral molecular clone. Running SPINE on this molecular clone leads to its division into sub-library fragments. SPINE will then generate inverse PCR primers containing BSMB1 sites. This will lead to the generation of a plasmid backbone flanked with BSMB1 sites and lacking the fragment of interest that we want to diversify. The diversity that we want to introduce will be through the form of these mutagenized oligo libraries that we can amplify specifically, and this will lead to this amplified sublibrary. These oligopools are also flanked with BSMB1 sites, and therefore we can mix these oligopools with this plasmid backbone into a Golden Gate assembly reaction, leading to the formation of a mutagenized plasmid library. This plasmid library will contain in this sublibrary fragment all of our variants of interest. We have to do this process iteratively at each sublibrary fragment that we want to modify. Since our variant of interest here today are insertions, we've decided to insert a sequence called an insertional handle. An insertional handle contains two outward-facing BSA1 sites. And therefore, once we have this insertional handle library, we can add any sequence of interest flanked with BSA1 sites and perform Golden Gate assembly. This will lead to the generation of a new insertional library with any sequence of interest. And so we can see how this insertional handle library is a tool to generate insertional libraries in a high throughput manner. So let's get started with the results. First, here I'm showing you the insertional handle library that I've produced. On the y-axis is the variant count, and on the x-axis is the amino acid position. The alternating gray and white boxes represent the different viral proteins, and the dashed black lines represent the division between the different sublibraries. And so we can see directly that we've obtained all of our variants of interest over the viral proteome. In addition, we can see the variant counts are very tightly distributed. And so this all suggests that we've built a balanced, saturated insertional library in the EVA71 proteome. And so this allows us to answer the questions that I asked before. The first one being, where are insertions tolerated in viral proteins? And how is that tolerance affected by the size and the amino acid composition? The first type of experiment that we wanted to do is to ask the tolerance of single amino acid insertions. And therefore, we built a plasmid library where here on the x-axis are all the possible single amino acid insertions, and on the y-axis is the variant count. And so we can see here that we've built a well-balanced and diverse library containing all these different single amino acid insertions. However, when we look here at the right and we use this plasmid library to generate virus and sequence that virus, we start to notice that certain single amino acid insertions are very depleted and certain single amino acid insertions are enriched. One type of amino acid that we expected to be depleted is the proline here, which is known to break secondary structures. However, this is really a naive and global type of analysis. This does not really answer one of the two scenarios. Either for these enriched amino acids, there are more insertion regions that are tolerated, or the second option is that there are, for all these amino acids, the same hotspot regions detected, but there is a differential tolerance between them. And so this is why I'm plotting here the most depleted amino acids against the most enriched amino acids over the viral protein. And so what we can see immediately in these variant counts is that insertions are tolerated or constrained only in specific hotspot regions in the viral protein. Comparing the most depleted amino acids and the most enriched amino acids, we can see that there is not a significant increase in hotspot regions. So therefore, this suggests that the second option is happening. And looking at this hotspot region in the structural protein in VP3, we can see that indeed this is the case. So comparing here arginine, the variant count at this hotspot region is much lower than the variant count for the amino acid insertion tyrosine. This is also a really superficial analysis. And so to reduce the dimensionality, multidimensionality of the data, we've decided to perform principal component analysis. And we've focused for now on the region between VP1 and 2A, where this cleavage site here separates the structural proteins from the replication proteins. And so what we could see directly from these principal plotting these principal components is that these were sufficient to separate the single amino acid insertions according to certain biochemical properties. And this was evident with arginine that has the highest isoelectric point. And to the right here, plotting the principal component scores, we can see that the variation in our data is best explained for that the variation is best explained in principal component 1 and 3 by the changes at upstream of the cleavage site. However, the variation in our data in principal component 2 is best explained at the end terminus of 2A. And so going back to this graph, we can see that this variation for insertional tolerance for glycine is best explained by the changes in tolerance at this region upstream of the cleavage site. While for arginine, this is more of a combination of changes at the cleavage site, but also at the end terminus of 2A. And so we can see how a principal component analysis is a powerful tool to explain the variation in our data. Another type of experiment that we can do is, instead of performing single amino acid insertions, we can insert a string of amino acids. And so here we've decided to insert different useful tags. And this is for two reasons. The first reason being that these useful tags are very well studied and have very well defined structures. And so they can explain the variation in our data. And second, once we identify these optimal sites, we can use them to study the viral life cycle. So to the right here, I'm plotting in these different blocks, the different insertion sequences across the viral protein. And what we can see evidently is that the larger the insert is, the less number of hotspot regions that are present. Additionally, we start to see certain patterns. For example, at the VP1 and 2A cleavage site, this is apparently a conserved hotspot regions for all different types of insertions. However, looking at other viral proteins, we start to notice that hotspot regions can drastically vary with different insertion sequences. And so this means that the amino acid sequence itself can affect the hotspot region that is tolerated. In addition, when we compare the secondary structures of these insertion sequences, and here I'm comparing the alpha tag, which is known to form a stable alpha helix, to these insertion sequences that form linear epitopes, we start to see a remarkable different insertional tolerance profiles. And so this means that not only the sequence itself is important, but also the secondary structure of the insertion is important. And so then we've used these type of optimal sites to generate recombinant viruses containing different useful tags. And I've done this for many different useful tags, but here I'm just showing the 2A replication protein fused to the amnion green fluorescent protein. And we were able to rescue virus and passage it several times, and it's stable in the viral genome. And so then this leads me to the second part of my talk, where I want to discuss where are these tolerated insertions localized on the structure of the viral proteins, and if these regions are conserved throughout viral evolution. To answer this, we are going to plot the insertion counts on the structures of the viral proteins. The color code here in blue are very constrained for insertions, while in red means that it's very tolerant for insertions. Here, I'm plotting the insertion counts on the capsid pentamer. In black here is the receptor binding domain. And we can see that this region is very constrained for insertions. However, we start to see some surface exposed regions that are very tolerant to insertions and that are present, interestingly, at the same axis. Rotating the structure allows us to look at different regions, and we could see that the internal part of the capsid, which comes in contact with the viral genome, is pretty much constrained for insertions. And this makes sense in terms of its interactions with the viral genome. We can do this type of analysis for other viral proteins. For example, here, I'm showing the structure of the 2A protease, which is the most well-described enzyme of the virus and has this active site here called the catalytic triad. Now we can see next to the active site that this region is very constrained to insertions. However, if we start to rotate the structure, we start to see regions that can tolerate insertions. And this allows us to understand the logic behind insertional tolerance in certain viral proteins. The last piece of data that I want to show you is what is the role of these insertions in the context of enterovirus evolution. And here I'm focusing on the structural protein VP3, where I'm showing the structure of VP3, and I'm mapping here in the colors the conservation scale. So in green means that these regions are very variable, and in red are regions that are very conserved. And we can see that regions are very variable are often surface exposed. Comparing this type of data with our insertional screen, we could see that this loop here in VP3 is very well tolerant for insertions. But interestingly, it's also very variable for non-synonymous mutations. And so this suggests that this region is in general very mutationally robust, tolerating amino acid changes, but also insertions. And so this sheds the light on the role of insertions in enterovirus evolution. And so I want to finish this talk by concluding there are mainly two advances in this work. The first one being a technological advance, where we've shown and we've adapted SPINE, where it provides an efficient way to perform deep insertional screening in viral genomes. We've also developed a mapping script that I haven't talked about much, but allows us to identify the position of large insertions, even from noisy sequencing data such as nanopore data. For the scientific advances, we've seen that EVA71 proteome in general is constrained to insertions to specific hotspot regions. And this tolerance can vary with the changes in the size, the composition, and also the secondary structures of the amino acid sequence. We've also identified optimal sites for useful tags in the viral genome, and these will be really instrumental tools for future molecular virology studies on this virus. And finally, with respect to the future work of the project, the short-term work will be mainly that we want to complete the mutational landscape investigated for EVA71 by performing deep deletional scanning and deep mutational scanning. We also want to develop phylogenetic-based methods that will incorporate fitness effects of mutations generated by the deep mutational scanning. For the long-term work, we want to study how selection pressures such as temperature, immune or antiviral pressures can modulate the fitness effects of mutations in general. We also want to start moving away from cell lines and use other relevant models such as human intestinal and airway organoids. Finally, I would like to acknowledge the lab that I've done the study in, called Quantitative Virology or Evolution Unit, or QView, which is really a nice place to do science and study RNA virus evolution. I would like to thank Patrick Dolan, my PI, for helping with the development of the computational pipelines that were necessary for this project and for his constant support. And finally, I would like to thank BEI because they are a repository that provided us with plasmids and also viruses for these studies. And thank you. And if you have any questions, feel free to ask. Thanks.
Thank you, William, for the amazing talk and the fascinating research. While we wait for questions of the public, we have some questions. So Diego is asking, for the people that don't have a strong background in virology, if you could give us a little bit of background of understanding how viral insertions are generated. Does it have anything to do with poor fidelity polymerases? Or I would add, does it have any correlation with transpositions or transposon viral evolution?
So yeah, that's a very good question. It has to do mainly with really specifically for positive single-strand RNA viruses with the RNA-dependent RNA polymerase, which can sometimes perform template switching. And so this is one mechanism how insertions can be produced. And then recombination, which is very well described in enteroviruses. For transposons, that's more specifically related to retroviruses, right? Which I'm really not talking about here. I'm really talking about viruses that always have positive strand RNA viral genomes all throughout their life cycle. But yeah, that's a very good question.
Thank you. So we have a question from Didier Devore, and he's asking, how did you select which PCs were used for the PCA analysis? He says, I didn't see PC1 involved, but only PC2 and PC3.
Yeah, we just decided here to plot PC2 and PC3 because we wanted to see this. We wanted to show these interesting differences that we saw regarding arginine and also glycine. But we've also done the plots for PC1, and definitely when we publish this work, we'll be showing more principal components. But yeah, we've decided on the 3 or 4 first principal components that explain most of the variation in data, right? So yeah, we've also done this type of analysis on the useful tags, and this also was interesting. So we're still like finishing up on this type of analysis.
Cool, thank you. Do we have time for more questions? Maybe one? Yeah. Okay, so I have actually 2 questions. So one is that I noticed that there is kind of a hotspot. I think you mentioned a little bit about the hotspots of regions that are enriched and depleted, and they are roughly at the edges of the viral protein. Do you know why you see right there at the beginning and the end of the viral protein these hotspots?
Yes, certainly for certain viral proteins, yes, where it's been well described. So for example, you've mentioned this, but if we look at the start of the genome, we see no hotspot regions. So at the start of after the start codon. And this is because, for example, this disrupts this small viral protein called VP4, and also can disrupt an upstream open reading frame. And so this variation that we see can be related sometimes to the functions. And Certainly this, for example, VP1-2A region cleavage site is a region which is very well tolerant for insertions for the capsids, because this is where it would probably get cleaved off the other parts of the viral proteins. So probably relating to cleavage sites. And even if these regions are tolerated, but also there are internal regions, which when you look at these structures, I think these are the probably the more fascinating ones to look at, right? Because N-terminus or C-terminus of proteins in general, this is where people put tags, right?
Yeah.
But yeah, that's
nice. So my second question was that I thought it was so interesting, the analysis that you did with the fitness scores and the entropy. So I was wondering if you also look at stability or predicted stability, and it has the same correlation with the entropy and the fitness.
So can you ask that question again? Is that relating to the last slides that I showed?
Yeah, you showed a slide with entropy and fitness, and it's highly correlated, the peak. So I was wondering if that same thing happens to stability of that mutant, if you did any analysis with the predicted stability of certain inserts.
Okay, so sorry, this is like that slide with the entropy is not my data. It's published work that was done for deep mutational scanning. So it's non-synonymous mutations and not insertions. But I believe that they've done stability. They've looked at stability of these, if these like change the structure of the virus or not. But yeah, this is not my data, sorry.
No problem. Thank you so much for your talk. And now we can go ahead to our second speaker of the day,